Hi, my name is Sharifan Amalina, and today I'll be presenting on behalf of my team from UBM on our research entitled The Analysis of Chemical Composition, Bioactive Compounds, and Antioxidant Activity of Malaysian Stainless B Propolis Water Extracts. So, propolis generally contain resins, waxes, essential oils, pollens, and also impurities. And propolis extracts are known to contain numerous beneficial bioactive compounds and biological actions that can be utilized in the pharmaceutical and health sectors. However, there's very limited reports or studies available, particularly on the stainless bee propolis in comparison to the honeybees in Malaysia, and water is also used instead of alcohols as the extraction solvent in this study because past reports had shown that there are unfavorable side effects when consuming or utilizing the alcoholic extracts. So this study aimed to investigate the total phenolics, flavonoids, and antioxidant activities of 3 Malaysian stainless bee propolis extracts, and the chemical groups and bioactive compounds were also successfully identified using the GCMS analysis. So in this study, propolis from Heterotrigona fimbriata, Tetragona apicalis, and Tetragona bilhami were extracted using water as solvent and underwent the freeze drying process for chemical analysis. And then the total phenolics, flavonoids, and DPPH scavenging assays of the samples were also determined, and GCMS analysis was carried out to identify any bioactive compounds present in them. And all of this analysis was done in triplicates, and one-way ANOVA and Pearson's coefficient correlations were carried out for the statistical analysis. So Table 1 and Table 2 showed the results obtained from the chemical and statistical analysis of the samples. Overall, it can be seen that H. fimbriata contained the highest concentration of phenolics, flavonoids, and antioxidant activities among all of the samples, and it can also be seen that there is a strong correlation between the concentration of polyphenols with the antioxidant activities exhibited by them. And figure 1 showed the percentages of major chemical compounds identified in all of the samples using GCMS analysis. So the findings from this study may differ from other studies on stainless bee propolis due to differences in bee species, location, plant preferences, and vegetations pollinated by the bees. And the solvent used during the extraction process is also important, and reports had shown water extracts to be more environmentally friendly, safe, and biocompatible for utilization and consumption in the health sectors. So in conclusion, the Malaysian stainless bee propolis used in the study contain bioactive components and antioxidants that could potentially be important for utilization in the health and pharmaceutical sectors. And hopefully this study will be able to encourage further research and investigation on stainless bee propolis to enhance their understanding and application, especially for our local stainless bee propolis in the near future. Thank you.
Hello everyone, my name is Nosha Zona Sahira from University Putra Malaysia. Today I'm going to present on the antioxidant capacity and in vitro antidiabetic activity of Cynomenstra cauliflower fruit. Cynomenstra cauliflower fruit, or normally known as nam-nam or buah katak puru, is a native in Malaysia. It is known by its high nutritive and medicinal value. The objective of this study is to evaluate the antioxidant capacity and in vitro antidiabetic activity of Cynomenstra cauliflower fruit extracted with different drying method and ethanol ratio. And the problem statement of this study is the optimization of the drying method and solvent extraction process is crucial to produce high quality functional ingredient. The extraction of this sample consists of 6 stages, which is drying, with different drying method, oven drying and freeze drying, grounding, and extraction with different ethanol ratio, which is 100%, 50%, and 0%, and then sonication for 1 hour, purification and lyophilization. And then the crude extract was subjected to 2 types of analysis. First is antioxidant capacity, consists of total phenolic content, total flavonoid content, DPPH, and ferrite reducing antioxidant power. And for in vitro antidiabetic activity, alpha amylase and alpha glucosidase inhibition assay was determined. From the results, we can observe that 50% freeze-dried extract show the highest TPC and TFC value. However, there is no significant difference in terms of drying method used. Then, we can observe that 50% freeze-dried extracts exhibit the strongest reducing power and also the strongest scavenging assay, whereas the IC50 value is below than 100 microgram per ml, which indicate that lower IC50 value indicate that the strongest antioxidant capacity. And for in vitro antidiabetic activity, alpha-glucosidase assay and alpha-amylase assay show the similar pattern, whereas 50% freeze-dried extract show the lowest IC50 value. For the conclusion, the best solvent extraction and drying method for antioxidant capacity and in vitro antidiabetic activity is 50% ethanol and freeze drying respectively. For the recommendation, further study can be conducted to identify the active metabolite to show the potential of the extract as the natural antioxidant and antidiabetic agent in food industry. That's all from me. Thank you.
Hello, my name is Mary, and the title of my poster is Toxicity Assessment of Oils and Extract from 4 Cymbopogon species. Cymbopogon nudus and Cymbopogon winterianus are commonly known as citronella grass or serai wangi. They are used in perfumery and cosmetic products. Cymbopogon citratus is the lemongrass or serai that we use in cooking. Cymbopogon martini is cultivated for its aromatic oil that is sweet and rose-like. These 4 Cymbopogon species look similar and may cause misidentification that could lead to toxicity. The aim of this study is to assess the toxicity of the oils and water extracts from the leaf of these 4 species. Phototoxicity was conducted based on OECD 2004 with modification. For non-phototoxic sample, example L-histidine, the dose-response curve are almost similar whether irradiated, the red line, or not irradiated, the blue line, with 5 joule per cm square UVA. Samples that are phototoxic, such as chlorpromazine, irradiated dose-response curve shows lower viability than the non-irradiated counterpart. Another indication is the mean photo effect and photoirradiation factor. Non-phototoxic sample has MPE value of less than 0.1 and PIF value of less than 2. Results showed that all the oils and water extracts of these 4 species were not phototoxic. Cytotoxicity was conducted on Vero, that is kidney cells, WRL68, liver cells, and BARB3T3 clone A31 fibroblast cells. After the cells were exposed to the test sample for 24 hours, viability was assessed by the MTT assay. Higher IC50 value is an indication of the samples being less cytotoxic and vice versa. The highest concentration tested was 500 microgram per ml. Overall, the water extract appeared to be less cytotoxic than the oils. In conclusion, all the oils and water extracts were not phototoxic but showed varying degree of cytotoxicity. This project is part of the FRGS project funded by the Ministry of Education Malaysia. Thank you.
Good day, everyone. I'm Poyen from UDKL. Today, I would like to share with you my study result, which is about the SAR of curcumin derivative in triple-negative breast cancer cells. Curcumin derivatives are structurally modified compound based on curcumin, which is a naturally compound extracted from root of turmeric. The aim of the structurally modification is to enhance anti-cancer effects and also selectivity against cancer cells. The strategy that we adopt in this study is to change symmetric to the asymmetric structure, to shorten the 7-carbon linker to 5-carbon, to introduce cyclic ring as a core linker, and to remove carbonyl group from the structure. There were a total of 38 compounds has been successfully synthesized and characterized. We screen this compound at 10 micromolar on TNBC and also non-TNBC cells. We determine IC50 and selectivity index using cancerous cell and non-cancerous cells. Gemcitabine was used as a standard drug, while curcumin was used as a parent compound. We highlight few important results here. Here showing the heat map of IC50 and selectivity index. The stronger green color indicating the compounds are more active and more selective to the cancerous cell. As you can see here, gemcitabine as a standard drug was highly potent with low IC50 value. But gemcitabine was also more toxic to the non-cancerous cell. In comparison to the curcumin, the potency is comparatively low. Similar, this compound was less toxic to the non-cancerous cell. For our compound 1 until 3, introducing one additional binding site improved growth inhibition effects and also selectivity. But introducing 2 binding sites caused toxicity to the non-cancerous cell. For the dicarbonyl series, only compound 6 show anti-cancer effects from the total 11 compounds synthesized. When the CF3 was replaced with the NO2, which has similar characteristics, the anti-cancer activity is totally abolished, showing that CF3 is unique to the anti-cancer activities of dicarbonyl group. When we introduce heterocyclic ring to the compound 11 until 14, all compounds show 10-fold more active than curcumin. Specifically to compound 12 and 13, which are more potent and also more selective to the most aggressive TNBC MDA cells, whereas other compounds were highly active and selective to the non-TNBC MCF cell and also TNBC HCC cell. In conclusion, introduced cyclic and asymmetrical structure of the curcumin derivative could improve anti-cancer activity and selectivity compared to the curcumin. I would like to thank to the FRGS and also UKM grant for these studies. Thank you very much for listening to my presentation. Thank you.
Assalamualaikum and hi to everyone. I'm Nur Ayuni. Today I'll be presenting my research poster entitled The Identification of Chemical Composition and Antioxidant Activity of Malaysian Stingless B Propolis using ethanol extraction. Firstly, stingless B. Stingless B, as the name suggests, they are lack of sting and well adapted in the tropical countries. In Malaysia, there are approximately 30 species of stingless bee that have been reported residing in the virgin forest and the rural area, which known locally as lebah kelulut. Next, propolis. Propolis is a natural, resinous and waxy product that is produced by mixing the bee wax and the resin collected from the various part of plant by the bee worker. Propolis also usually composed of 50% resin, 30% waxes, 10% essential oil, 5% pollen and 5% of organic compounds. Other than that, propolis also uniquely capable to exhibit tremendous medicinal properties such as antimicrobial, antioxidant, antifungal and many more. Furthermore, in this study also we'll be focusing more on the propolis extraction using ethanol as a solvent. Why? Because alcohol can provide higher range of bioactive concentration compared to non-ethanolic solvents such as water and oil. Therefore, this study aims first to determine the total phenolic and flavonoid content using the UV spectrophotometer as well as the bioactive constituents using the DCMS analysis from 3 different Malaysian stingless bee propolis. Second is to determine the correlation between the phenolic and flavonoid content with the antioxidant activity of each propolis sample. Moving on to the methodology, first step is the propolis collection, second is the propolis ethanolic extraction preparation, the third one is the UV spectrophotometer analysis to analyze the TPC, which is the total phenolic content, total flavonoid content as well as the DPPH free radical scavenging activity. Next is the GCMS analysis, in which to determine the bioactive constituents from several major groups. And the last one is the statistical analysis used in the study, which is one-way ANOVA as well as the Pearson correlation coefficient. So the result obtained from this study is Trigona apicalis recorded the highest total phenolic content, while Homo trigona fimbriata recorded the highest total flavonoid content as shown in table 1. Other than that, Homo trigona fimbriata also recorded the highest percentage of DPPA scavenging activity and the lowest IC50 value as compared to the standard solution, which is the ascorbic acid as shown in table 2. Besides, it is also noted that there is a strong correlation between the total phenolic and flavonoid content with the antioxidant activity of each propolis extracts. Moving on to the next figure, figure 1. Figure 1 shows the bioactive constituents analyzed using the GCMS analysis, and over 30 constituents detected and categorized into 6 major groups such as terpenoid, ketone, carboxylic acids, hydrocarbon, alkaloids, and other constituents. However, the concentration of the bioactive and the polyphenol components may vary, depends on their floral preference, bee species, collecting seasons, and the solvent used during the extraction. In conclusion, this study may be able to encourage further studies on the bioactive constituents of stingless bee propolis, which may lead to a new discoveries of its composition and possible application.
Greetings! My name is Mas Fatika binti Rizazman, and I will be sharing the findings from my research project entitled Phytochemicals from the Stem Bark of Calophyllum canum. Calophyllum canum, or Binthamomera, is an evergreen tree growing from 7.5 to 36 meter tall. The size ranging from medium to large trees, and it grows in well-drained ground as well as pit swamps up to 1,200 meter altitude. Calophyllum canum is native to Peninsular Malaysia, Borneo and Sumatra. The latex is used in tuba fishing by the old folks, and wood is the main source of timber, especially in Sarawak. Previous studies have reported the biological activities of the Calophyllum species, such as anti-cancer, antibacterial, and antioxidant. Next, the objective of this study is to isolate and characterize the chemical constituents from the stem bark of Calophyllum canum. The procedures are the common method used by the natural product researchers. The stem bark was ground and macerated with chloroform. Then the chloroform extract was concentrated in vacuum and subjected to vacuum column chromatography for fractionation. The fractions of the similar chemical profile were combined and subjected to column chromatography for isolation and purification processes to obtain the pure compounds. As illustrated, this is the summary of the isolation and purification work done for the stem barks of Calophyllum canum. A combination of chromatographies such as vacuum column, gravity column, and radial chromatographies were applied. Based on our findings, trapeziphali-xanthone, anani-xanthone, e-xanthone, and fridolin were successfully isolated from the chloroform extract of this Calophyllum species. As a conclusion, the phytochemical study on the stem bark of Calophyllum canum yielded 3 xanthone derivatives, which are trapeziphali-xanthone, anani-xanthone, e-xanthone, and a common tritopene, which is fridolin. These are the references of the information regarding the studied plant of this species. Last but not least, we would like to acknowledge the SRDC and UITM Sarawak for the financial assistance and support given throughout the research conducted on genus Calophyllum.
Assalamualaikum and good day everybody. My name is Nurdiana Betty Ibrahim. I'm student at UniKL Royal College of Medicine Perak. Today I would like to present about in vitro antioxidant and photoprotective activity of the Graptofilin leaf extract. Recently, natural substance extract from the medicine plant has been used in medicines and cosmetics due to their absorption in the ultraviolet region and their antioxidant activity against skin damage. Phytochemical screening of the G-peptide consists of the tannin, flavonoid, saponin, alkaloid, glycoside, peptide, formic acid, steroid, and alcohol. The present study aimed to evaluate of the antioxidant and photoprotective activity of the different solvent extract of the G-peptide leaf. There were four steps in method of the study. Firstly, the dry powder leaf were extracted separately in ethanol and methanol by saltlet extraction method. Secondly, total phenolic content and total tannic content were determined by follicular culture method, while the total flavonoid content was by aluminum chloride method. In vitro antioxidant activity of the extract were determined by three different spectrophotometry methods: DPPH assay, ABTS assay, and hydrogen peroxide assay, and compared with the ascorbic acid as a standard. Lastly, the sun protection factor, all of extract were determined using UV spectrophotometer from the 290 nanometer to 320 nanometer at various concentration. From the findings, the highest total phenolic content was found in methanol extract, while the total flavonoid content and total tannic content exhibit highest in ethanol extract. In DPPH assay, ABTS assay, and hydrogen peroxide assay for the both solvent extract, ethanol extract was found good 50% inhibition concentration compared with the methanol extract. The result DPPH assay was 31.17 microgram per ml. ABTS assay was 70.96 microgram per ml and hydrogen peroxide assay was 10 microgram per ml. However, ascorbic acid was found potent antioxidant activity compared with the all extract. The highest SPS value was found in methanol extract with the result 27.67 compared with the ethanol extract in a concentration dependent manner. This finding revealed that the leaf extract of the G-peptide possesses a great antioxidant and photoprotective potential which can be used as a natural active ingredient in sunscreen preparation due to the presence of the flavonoid, tannin, and phenolic compound. Lastly, we gratefully thank UniKL for providing STRG grant and financial support during the course of the research work. Thank you.
Assalamualaikum and greetings to everyone. It is a great pleasure to participate in this prestigious symposium. Today, I am Nur Maria Ulfa binti Seruji. I will show you our research findings as a knowledge expansion in the Calophyllum genus. In this research, we have worked on the undiscovered Calophyllum microbiome. It is an interesting study since this is one of the untold stories of Calophyllum genus. We have worked on the phytochemical studies, where the extracts were treated with various chromatography techniques for the isolation of the compounds, which then identified using advanced spectroscopic methods. Besides that, all extracts were tested for its cytotoxicity against the cervical cancer cell lines and human prostate cancer cell lines using the entity assay protocol. This effort leads to a success where four non-compounds were isolated and identified as trapezifolate xanthones, twice xanthones, beta-sitosterol, and fridolin. Moving on to the cytotoxic activity results, there were three drugs were used as positive controls, and they were doxorubicin hydrochloride, cisplatin, and fluorosilver. Based on the data obtained for the cervical cancer cell lines, all the extracts exhibited cytotoxicity below 1.2 milligram per ml of IC50 value. The chloroform extract showed the highest activity after 24 hours, but ethyl acetate demonstrated a better activity after 48 hours. The chloroform extract activity was found 15-fold stronger compared to fluorouracil drug after 24 hours. In addition, ethyl acetate extract showed a higher cytotoxicity than fluorouracil drug, but weaker than the other two controls. As for the prostate cancer cell lines, all extracts showed IC50 value of below 1.0 mg per ml at 48 hours. The ethyl acetate extract is the most potent against this cell line with IC50 value of 0.058 mg per ml in 48 hours. Moreover, both chloroform and ethyl acetate extracts exhibited more potent cytotoxic activity as compared to fluorouracil drug, but weaker than other two controls. Thus, from these results, it is thought that further study should be done on clavulin recurvatin, where these findings will be an additional contribution to the chemotaxonomy of clavulin genus. Well, that's all from me now, but before I end, on behalf of my team, I would like to sincerely thank Sarawak's Research Development Council and YTM for financial support throughout this research. Thank you and stay safe.
Assalamualaikum. The title for my poster presentation is Bioactive Extract and Compound from Ardisia crispa root exert anti-cancer effects on colorectal cancer by impeding angiogenesis and metastasis in vitro. As for the introduction, the ACRH and BQ have been reported to exert promising anti-inflammatory and anti-angiogenic activities, and angiogenesis has been known to play a crucial role in the progression of colorectal cancer. Hence, the objective is to investigate the potential of ACRH and BQ in impeding angiogenesis and metastasis in colorectal cancer cell lines. For the methodology, two types of CRC cells are being used. The first one is the metastatic type, which is LoVo cell, and the second one is the primary tumor HCT116 cell lines. Okay. For the method, cell viability assay was conducted, followed by annex in 5-bit CPI staining, scratch assay, invasion and migration assay, and finally all data obtained was collected and analyzed using one-way ANOVA and followed by post-hoc analysis. For the result, the viability of the cells were evaluated using MTT assay, and results in figure A showed that ACRH exerts similar cytotoxic effect on both HCT116 and LoVo cell lines with IC50 for HCT116 at 1.9 microgram per ml, and for LoVo cells the IC50 was 1.5 microgram per ml. On the other hand, figure B showed BQ concentration where IC50 for HCT116 was set at 2.1 microgram per ml and for LoVo 2.4 microgram per ml. Subsequent experiments were then conducted with HCT116 and LoVo cells treated with HCT and with ACRH and BQ at concentration of 2.0, 0.2 and 0.02 microgram per ml. Figure C and D showed apoptotic activity that was measured using flow cytometry, in which the results notably correlate with cell viability with increased apoptotic rate of both HCT116 and LoVo cells in a dose-dependent manner. On the other hand, figure E and F showed scratch assay resulted in significant inhibitory effect on the proliferative and migratory ability, particularly of LoVo cells treated with BQ at 2.0 microgram per ml, with only 36% of cell migration. In addition, for figure G and H, it shows migration and invasion assay using transwell Boyden chamber that indicated that after 24 hours of treatment with ACRH and BQ, exert significant effects in inhibiting the mobility of both HCT116 and LoVo cells. And finally, although HCT116 cells treated with ACRH at 2.0 microgram per ml showed to have the least migrated or 0.9% and the least invaded cells 1%, the overall treatment of ACRH and BQ at all concentrations are observed to be more effective in inhibiting LoVo cells compared to HCT116 cells. Finally, these current results suggest the promising potential of ACRH and BQ as a valuable candidate for the development of colorectal cancer therapy. Hence, future works will involve molecular studies of proteins and identification of biochemical pathways affected by treatment with cells with affected by treatments of cells with ACRH and BQ. Thank you very much.
My name is Muhammad Izzuddin bin Nuzul, and in this video, I will be giving a brief explanation about my research entitled Effect of Different Extraction Techniques on Total Phenolic and Flavonoid Contents and Antioxidant Activity of Bambusa bichayana mudro. Introduction. Bamboo is commonly known as a material used in construction and furniture. In this research, I am focusing on one of the bamboo species known as Bambusa bichayana. Bambusa bichayana is found in the tropical region of Southeast Asia, with its height ranges from 7 to 16 meters with internal lengths of 34 to 41 centimeters, comb diameters of 6 to 10 centimeters, and wall thickness of 1.5 to 2 centimeters. Nowadays, many studies were focused on its potential as a source of nutraceutical and cosmeceutical for healthcare products. Research has shown the potential of bamboo use as food and healthcare-based products. Objective. The study is conducted to investigate different types of extraction techniques and solvents in determining the total phenolic and flavonoid content as well as free radical scavenging activity in the extract of the comb of Bambusa bichayana mudro. Methodology. The comb powder was extracted in 3 different solvents: ethanol, methanol, and water, with 3 different extraction techniques, which are maceration, successive extraction, and ultrasonic assisted extraction. For ultrasonic assisted extraction, it was further investigated at different time intervals, which are 20, 40, and 60 minutes. The analysis of the total phenolic content, total flavonoid content, and the DPPH antioxidant assay were carried out by using UV-vis spectral photometer.
Results. From the table, it shows that the methanol extract extracted using successive extraction has the highest total phenolic content, highest total flavonoid content, and possess more potent antioxidant activities. And as the conclusion, successive extraction is potentially the most suitable method to be used for extraction of bioactive compound. Hence, this research provided scientific evidence for the research of bioactive compounds and the exploitation of bamboo as food products.
Hello, I'm Geeta from Forest Research Institute, Malaysia. My poster title is In Vitro Antistaphylococcal Activities of a Dimeric Sesquiterpenoid and its Effect Against Fusidic Acid-Resistant MRSA. Fusidic acid is widely used as monotherapy to treat superficial cutaneous infections and infected traumatic wounds such as surgical diabetes and burn wounds. When combined with selected antibiotics, fusidic acid has proven effective for difficult to treat methicillin-resistant Staphylococcus aureus or MRSA, where first-line topical antibiotics such as mupirocin failed. However, its widespread use resulted in global emergence of fusidic acid-resistant MRSA or FRSA. The objective of this study is to evaluate antistaphylococcal activities of a dimeric sesquiterpenoid compound coded as DS produced by a wild Basidiomyces fungal strain coded FRIM550 and to study its effect against FRSA. First, the fungal culture is grown in bioreactor and culture broth extracted using solvents, followed by bioactivity-guided isolation of DS, and its structure was elucidated. Then, MIC values of DS were determined against a panel of 5 MRSA isolates, including 2 clinical FRSA using broth microdilution assay. MIC was defined as the lowest compound concentration required to inhibit visible bacterial growth. DS activity was compared with the last resort systemic anti-MRSA drug vancomycin and 2 common topical anti-staphylococcal agents that are fusidic acid and gentamicin. Table 1 showed that DS has MICs ranging from 3.9 to 7.8 micrograms per mil against all MRSA and FRSA isolates. This is close to the MIC values of vancomycin. Interestingly, DS was found to be more effective than fusidic acid and gentamicin against the FRSA. Next, DS and vancomycin were subjected to a time kill assay using a reference MRSA isolate. Bactericidal level is indicated by the dashed lines in figure 1. Both DS and vancomycin displayed bactericidal effects against MRSA at all test concentrations. Time kill curves showed that DS was far more rapid compared to vancomycin in its MRSA killing activity, that is within 1 hour, in a time-dependent manner. Lastly, DS was studied for its effect when combined with topical anti-staph agents, fusidic acid and gentamicin, against test MRSA and FRSA isolates. The checkerboard assay method was used. Figure 2 showed that no antagonism was observed for either of the combinations against all test MRSA and FRSA. In fact, DS significantly reduced the MIC of fusidic acid and gentamicin against FRSA. The DS fusidic acid combination was synergistic against both clinical FRSA. Synergy and partial synergy were also observed for the DS gentamicin combination against FRSA. Interestingly, the combination with fusidic acid greatly reduced MIC of DS against both FRSA isolates by 8 to 16 folds. In conclusion, the results strongly suggest that DS has the potential to be studied further as a new class of topical antibacterial agent for treating drug-resistant MRSA skin infections. Thank you.
Assalamualaikum and greetings to esteemed judges and audiences of this presentation. I'm Huda Zulfa binti Muhammad Zainuddin, PhD candidate from UNIZA. I will be presenting a research project entitled Anti-Proliferative Activities of Chrysanthemum morifolium flower aqueous extract against human chronic myeloid leukemia K562 cell lines. For the introduction, Chrysanthemum morifolium, a popular herb in Asia, is now discovered as a source for cancer treatment. So in this study, we investigate the anti-proliferative activities of this plant extract against K562 cell lines. So in materials and methods, we prepared crude aqueous extract from dried flower of this plant using maceration technique and tested against K562 cells via MTT assay and cell morphological observation using phase contrast microscopy in 24, 48, and 72 hours incubation period. For the results, in figure 1, significant cell viability reduction of K562 cells for all incubation periods, and IC50 value of the extract is shown in table 1. For table 2, or figure 2, showed the increasing apoptosis phenomena in treated K562 cells as incubation time increases. So these results showed that the extract significantly inhibit proliferation of K562 cell lines and induce apoptosis in a time and dose dependent manner. Thus, as a conclusion, Chrysanthemum morifolium could serve as an alternative drug candidate for chronic myeloid leukemia treatment. Lastly, we would like to thank UNIZA and Ministry of Higher Education Malaysia for funding this research. Thank you for your viewing.
Thank you for accepting our paper as poster presentation in ASOMPS 2021. The title of our paper is Trapezium folizenton as a chemotaxonomy marker in the genus Calophyllum. This is a review paper of the phytochemical studies conducted on the genus suggests the role of trapezifolizanthone as chemotaxonomy marker that aid in botanical identification of respective calophyllum species. In addition, trapezifolizanthone exhibit a various potent pharmacological activities to preclinical studies. This is the tree of calophyllum and the structure of calophyllum, trapezifolizanthone. So trapezifolizanthone is a premium perennial xanthone. The IUPAC name is 5,10-dihydroxy-2,2-dimethyl 3-methyl-buta-2-anyl, perennial 3,2-B, xanthan 6-on. So trapezifolizanthone was regarded as a common natural occurring compound, previously isolated from reported on calophyllum throughout the years. The genus calophyllum is not well studied until the discovery of potential HIV inhibitor calonolide A from Calophyllum lanigerum, which embarked the interest of the researchers in this genus to investigate the potential of phytochemicals as hit or lead compounds against various diseases in the development of the drugs. So table 1 shows the list of calophyllum species with trapezifolizanthone. This is the spectroscopic characterization of trapezifolizanthone. So it was isolated as yellow needle crystals or yellow prism with a melting point of 145 to 146 degrees Celsius. So the NMR spectra data of trapezifolizanthone is listed in table 2 based on Zama Shari et al. 2016. This is the we have proposed a plausible biosynthesis pathway for from compound 7, which is 1,3,5-trihydroxyxanthone, which undergo prenylation to form dexigartanin 8, and after that the compound 8 will undergo cyclization reaction to form perennial ring, which leads to the formation of trapezifolizanthone. SAR studies the relative position of hydroxy prenyl perennial group relatively at carbon 1 to 5 in xanthone nucleus plays a vital role in promoting inhibiting pharmacological activity. So in conclusion, in summary, the review highlighted the abundance of trapezifolizanthone and calophyllum species. So yeah, this is the references, and we would like to acknowledge Sarawak Research Development Council SRDC and UiTM Sarawak for financial assistance and support given throughout the research conducted on genus calophyllum. Thank you.
Assalamualaikum and salam sejahtera. Today I'm going to present our research finding in vitro antiviral activity of plant extract from Clinicanthus nutans. Clinicanthus nutans is a shrub and found in deciduous forest. It has been traditionally used to treat various conditions. A recent study showed that C. nutan has significant antiviral activity against various viruses. HSV1 is a common pathogen caused cold sore or common cold and oral labial infection. The infections can be successfully treated using acyclovir and affiliated nucleoside analog, but the emergence of drug resistance has created barriers to the treatment, especially in immunocompromised patients. And this resistance is caused by mutations in either thymidine kinase or DNA polymerase gene. The study conducted based on two objectives: to determine cytotoxic concentration specific value of C. nutan and to evaluate the mode of action against HSV1 via 3 different assays. For the materials and methods, plant use is the methanol extract of C. nutans, cells and virus obtained from stock at Virology Laboratory UKM. Cytotoxicity test was performed using MTT assay. Antiviral assay was determined using plug assay, composed of post-treatment, pre-treatment, and the residual treatment. Post-treatment assay is a treatment for 48 hours post-absorption. Pre-treatment is a pre-treatment with extract for 24 hours prior to infection. The residual assay is a mixture of virus and plant extract directed to cell-free virus. For the results, for Figure 1, cytotoxic concentration specific value of C. nutan extract is determined as 1.625 mg per ml. Based on Figure 2, post-treatment show 80% plaque reduction at 1.25 mg per ml. In pre-treatment, more than 50% plaque reduction was achieved at a minimum concentration of 0.313 mg per ml. For biocidal assay, more than 50% plaque reduction was observed at a minimum concentration of 0.6 to 5 mg per ml. The effectiveness of C-Newton extract as an antiviral compound expressed as selectivity index, SI. And from the table, C-Newton extract has greater SI value in post-treatment followed by pre-treatment and biocidal. Any antimicrobial compound that has SI value higher than 10 ensures the potential to be developed as an agent of antiviral, and this indicates C-Newton has potential as antiviral agent. To conclude, our findings suggest that C-Newton extract exhibits potent antiviral activity in vitro and contains antiviral active compound and could be potential antiviral agent. These results can help further study in developing process of C-Newton as potential anti-HSV1 therapy. We would like to thank University Sultan Tanah Abidin UNISA for giving us permission to use research facilities and supporting in this research. That's all from me. Thank you.
We are live.
Good afternoon, everyone. Ladies and gentlemen, before we begin today's afternoon session, I would like to get your response on ongoing event. Please give your feedback regarding the quality of audio and visual of this conference to the chat box. To all of the speakers and participants in this virtual platform, in order to ensure the smoothness of this event, I would like to remind you kindly mute your phone and mic during all ongoing sessions. The conference local share, our invited speaker, distinguished guests, ladies and gentlemen. First of all, it is my pleasure to be the MC for the 17th Asian Symposium of Medicine on Medicinal Plants and Spices 2021, ASOMPS 2021, with the tagline of Contextualizing Natural Product Science for Societal Relevance. My name is Thiruvendan Karunagaran from University Science Malaysia, USM, representing the Malaysian Natural Product Society for today's afternoon session. On behalf of the organizer, we wish to extend our warmest welcome again. Selamat datang to all participants, guests of honor, delegates, representatives, and not forgotten, our beloved participants to the 17th ASOMPS 2021, which is first time being held as virtual. Organized by the Malaysian Natural Product Society, together with our co-organizer, Malaysian Institute of Chemistry, Forest Research Institute Malaysia, Phytochemical Society of Asia, and Japan Analytical Industry. Now, we are ready to begin with our second plenary session. It is my pleasure to invite the Honorable Member of Malaysian Natural Product Society, Datin Dr. Rohaya Ahmad, to introduce our next distinguished speaker. Welcoming Datin Dr. Rohaya Ahmad.
Assalamualaikum. Thank you, Mr. MC. Assalamualaikum and good afternoon. Today I have given the honor of introducing our second plenary speaker, Prof. Ataur Rahman. As a renowned organic chemist and an eminent scientist, Prof. Atta deserves a long introduction, so I hope you will bear with me as I highlight some of his achievements. Prof. Atta obtained his PhD in organic chemistry from King's College, University of Cambridge in 1968 and later joined University of Karachi, Pakistan. Throughout his career, Prof. Atta has received many internationally prestigious awards. He won the prestigious UNESCO Science Prize in 1999, and he was honored with the highest Siebel Award by the Austrian government in 2007. And later in 2013, he won the Einstein Professorship by Chinese Academy of Sciences in 2013. He has also been conferred several honorary doctorate degrees, including a Doctorate of Science from University of Cambridge and Bradford University, and a Doctorate of Education from Coventry University. Currently, he serves as the co-chair of the UN Committee on Science, Technology and Innovation, the president of Network Academies of Sciences of Islamic Countries, NASIC. He's also the patron-in-chief of the International Center for Chemical and Biological Sciences, University of Karachi, and he is also the editor of the encyclopedic series on natural products, Studies in Natural Product Chemistry, published by Elsevier, which now carries 68 volumes. Of course, he's also the editor-in-chief of several European chemistry journals. In terms of publications, Prof. Atta has to date published 1,267 international publications, including 50 international patents, and 83 PhD students have graduated under his supervision. Despite his outstanding achievement, at a personal level, Prof. Atta is a very kind and humble person and is always happy and excited and eager to share his knowledge and expertise. Today, Prof. Atta will deliver a lecture entitled Efficient Approaches of Drug Discovery from Medicinal Plants and Catalytic Biotransformation. He could not be with us live, but he has graciously prepared a video for us. Without further ado, let us now watch and listen to the plenary lecture from Prof. Ataur Rahman. Thank you.
Good afternoon, everyone. Well, it's a pleasure and a privilege for me to be able to present this lecture, which will be focused on some of the exciting work that we have been doing in the field of natural product chemistry. And in the end, I will also talk to you a little bit about how innovations are driving economies and what we have been trying to do in Pakistan. So my lecture will be focused on, firstly, some areas of neuroscience in which we have been involved in. In Pakistan, we have about 6,000 different plant species, of which about 400 are medicinally important. If you look at the map of Pakistan, we find that in the south you have Karachi and the desert areas next to the seas at the bottom. And then as you proceed to the middle areas of Pakistan, these are the green fields, the so-called Punjab with 5 rivers flowing through. And then as you go to the north, you find the huge mountains, the Himalayas. And so you have a huge diversity of climate, minus 50 degrees up in the north, up in the top of the mountains, to plus 50 degrees centigrade in the summer in areas of the Sindh province. And with this huge geographical diversity, we find biodiversity also, varying natural products, natural plant materials, and varying structures of natural products on which we have focused over the last so many years. So at the HEJ Research Institute of Chemistry at Karachi University, where I work, over the years we have accumulated some 6,000 extracts from some 2,500 plants collected from different climatic zones of Pakistan. We have also been working on plant species from other regions such as Turkey, Jordan, Cameroon, and other areas of the world. And I have isolated many interesting natural products with exciting biological activities. But the focus of my the first part of my lecture will be on anti-epileptic natural products. Epilepsy is a neurological disorder, and it affects a large number of persons, and is characterized by unprovoked seizures. The causes can be genetic, they can be congenital defects, epilepsy can be caused by severe head injury or by ischemic injury or tumors. And the drugs available today are no better than they were 200 years ago. So there are no good drugs available against epilepsy. And the most common first-line treatments are drugs that block the sodium channels and suppress seizures, but do not actually cure the problem. So we carried out a nationwide ethnobotanical survey for identification of plants used in local folklore. And we found that there were a number of delphinium and aconitum species which had been used by folklore, by the local hakims. And one of the plants that we then extensively worked on is Delphinium denudatum. And the tubers of this plant are worn as a necklace by epileptic patients. This gentleman is not an epileptic patient, he's our lab boy, but he's just wearing it just because I asked him to wear it and just to get himself photographed. And but this is the way, and this gave us the clue that perhaps they may be small molecules and may therefore be aromatic principles or volatile principles present in the plant which are responsible for this activity. We have there are a number of tests that can be done. There's the electrical, the maximal electroshock test. There are chemical tests which involve subcutaneous injection. And there are and one can first of all by giving these cause sustained repetitive firing and then one can give these drugs and see the suppression of these sustained firing. by these drugs, which may be standard compounds or by plant extracts. And so this is the patch clamp facility that we have in our laboratory, in which we study the individual ion channels, and we can measure very weak currents of 10 to the minus 12th of an amp generated by the flow of various ions. And this then allows us to monitor how a neuron, so once one excites a neuron by giving it this weak current, it undergoes repetitive firing, uncontrolled repetitive firing, and then it is exposed to the standard compound or to the plant extract, or one of the fractions, and this tells us if there is any activity. And so, using these techniques, the bioassay guided isolation was carried out of these compounds that were isolated ultimately, and we found that the aqueous extract of some of the delphinium and aconitum species had significant anticonvulsant activity. And to cut a long story short, we isolated these very simple compounds initially, which were the isoxyletones. They are the E and Z isomers at the exocyclic double bond, and we're just drawn in different colors to show their potential bioactivity. And both of these isoletones showed the suppression of that they reduce the expression of the CFOS protein in kindled animals. CFOS actually belongs to the family of immediate early gene transcription factors, and its expression is known to increase with the neuronal activation. So we found that both these E and Z isoletones had potent activity to suppress CFOS protein expression. And similarly, the brain-derived neurotrophic factor, its expression was inhibited by both these isoxyletones. BDNF is a protein, is a member of the neurotrophin family of growth factors, and it is known that in epileptic patients, the level of BDNF rises. So these provided some clues regarding the mechanism of action of these compounds. And we carried out synthesis of a large number of derivatives, starting from isophorone using a Hornerwood-Wadworth-Emmons reaction. We prepared these derivatives, and then after hydrolysis and using we prepared various Weinreb amides, and then after Grignard reaction, ended up with a series of isoxyletone derivatives, such as compound number 5, and these are various compounds that were prepared for this through this synthesis, and we examined their biological activity in suppressing epilepsy, initially through using the patch clamp facility. This work was patented and has gone through phase 1 clinical trials and has entered phase 2 clinical trials. And we hope that something exciting will actually emerge from this. This led us into trying to understand what memory is all about. I have been thinking about this for quite some time. What are thoughts made of? Thoughts are not abstract. They are made of atoms and molecules. What atoms? What molecules? A lot is known about where thoughts are stored in the hypothalamus of the human brain. But how are they stored? What is the chemistry behind thought storage? Understanding of that can lead us to a better understanding of such neurodegenerative diseases as Alzheimer's, as Parkinson's, as epilepsy, and other neurodegenerative disorders. So when we look at the human brain, it's arguably the most complex object of our universe. There are 100 billion neurons in our brain. Each neuron is communicating with some 8 to 10,000 other neurons. So there are about 100 billion times 10,000 synaptic connections. So the process of thought, of thinking, of storing, of recalling is really one of the most remarkable features of nature. 100 billion neurons, about the same number as the number of stars in our galaxy, the Milky Way. When we think, certain parts of the brain light up. And here is an image of a functional MRI image of the brain when a person has been given a certain task of working so that when he's trying to resolve a problem, these MRI images, they show the changes in blood oxygen level which is taking place. And this is indicative of the brain activity. So it shows what parts of the brain are involved in the thought process. In the case of schizophrenia, if you see on the left side is a normal brain with the same part being lighted up, but schizophrenic patients are not able to perform in the same way. And on the right side, you see that only a certain part is being lighted up, the other parts are not being lighted up. So one can see that through functional MRI, but what is going on at the molecular level? That was the question. So what is the underlying chemical process that is involved? The models that exist internationally and have been proposed do not offer any chemical explanation. They only theorize that certain synapses may be modified by learning related activities which result in functional and structural stability. However, they do not address the basic issue of what molecules may be involved and how memory may be stored. So what we did, we proposed back in about 20 years ago, the hydrogen bonding patterns hypothesis. And we proposed that the main mechanism required to store memories could be the post-translational modification of synaptic proteins and nucleic acids through hydrogen bonding. So if you look at the glycoproteins present in the hypothalamus, glycoprotein, you have a protein backbone and you have sugars, various glucose, fructose, arabinose, xylose. And these sugars are bristling with hydroxyl groups. So it's like a velvet, a cloth on which you can emboss different patterns. So similarly, you have this natural velvet of hydroxyl groups. And these can be embossed through hydrogen bonding and patterns can be formed. So if you look at a watch, one pattern can be formed. If you look at another object, another pattern can be formed. And these patterns can then be stored. So the process of learning could involve formation of hydrogen bonds through bonding between hydroxyl groups with other hydroxyl groups present in the sugars or with the nucleic acids present nearby. And these could be intramolecular or intermolecular hydrogen bondings. So according to this hypothesis, the ability of the brain to gather, store, manipulate, and then retrieve the memory may be related to the reconstruction of these hydrogen bonded network of patterns. And this is the kind of pattern that we proposed through glycoconjugates formation. And these could be intramolecular or they could be intermolecular between different layers of hydrogen bonding. And so logic can then be looked at as a multidimensional overlay of these patterns. Each group of patterns may be associated with shape, color, fragrance, emotions, feelings of sadness or happiness. In fact, everything about us may be stored in the form of these patterns, and then your thought processes can then be considered as an interrelationship of these patterns through which the logic can be derived. So learning can be formation of these patterns, and forgetting can be the breaking of these patterns through the breaking of the hydrogen bonds. Long-term memories may also involve covalent bonding. So it was a few years ago, the patterns that we had proposed back about 20 years ago, some of these were discovered in microtubule lattices by some Canadian workers. And I've written a number of reviews, and we have a number of patents in this field. One of my reviews was in the Neuroscientist in 2014. Another one was in Current Medicinal Chemistry more recently. So functional MRI has not evolved to a point where you can see those molecular patterns, but I think this is still a hypothesis, but a very interesting and reasonable hypothesis on which work continues to go on. I will like to move now into the second phase of my lecture, and that is about aromatase inhibitors and how one can, how drug discovery can be carried out through modifying of existing drugs and developing new drugs from them. So what we were looking at different aromatase inhibitors, and what we decided to do was to select some of the existing aromatase inhibitors and then modify them using microbial transformation. And this means that we are starting with a privileged pharmacophore and then seeing whether we can improve its activity rather than starting at random with drugs. And so that multiplies our chances of discovering exciting molecules. The advantage of microbial transformations are that these occur in mild conditions. They are environmentally friendly, they're cost effective, these are single pot reactions, and these are regio and stereo selective. So you can introduce hydroxyl groups or epoxide groups or double bonds in selected molecules. And the process is fairly straightforward. I won't go into describing it. But coming to aromatase inhibitors, the most common cause of cancer-related deaths in women is breast cancer. And this is caused by the abnormal growth of cells in the form of cancer cells. And this can be an invasive ductal carcinoma or a lobular carcinoma. About 2/3 of the pre-menopausal and 3/4 of the post-menopausal breast cancers are caused by estrogen-dependent, the estrogen receptor positive ER+. And so if we deprive estrogen, this can be a way of inhibiting the progression of hormone-dependent cancers. The enzyme aromatase mediates in the production of estrogen. And so if you can inhibit aromatase with the suitable molecules, this could be an interesting treatment of estrogen-dependent cancers. So what we decided to do was to take various aromatase inhibitors and modify them. Now, what does aromatase do? If you look at, for instance, testosterone, the use of this enzyme results in decarboxylation of the methyl group between rings A and B, and aromatization of ring A, resulting in the formation of estradiol. And so this, and one can do this with various types of inhibitors. So we started with active molecules such as testosterone, which is shown on your slide. And using testosterone, we subjected to biotransformation and identified using various fungi, identified various molecules that were formed from hydroxylation. And if you look at, for instance, the compound which is formed, one of the compounds which is formed through hydroxylation here, it is the starting material has an IC50 of 1350 nanomicrons, but this product has an IC50 of only about 8.6, so it is about 40 to 50 times more active than the starting compound that was used. The structure of the products were confirmed through X-ray crystallography. Similarly, the atomistane was used. This is again a very potent aromatase inhibitor, and this was again subjected to fungal transformation using Fusarium linnae this time. And the compound that is shown at the bottom left of the slide is much more active than the starting material. It has undergone a hydroxylation. Another product formed is the one which is highlighted in yellow, which is again more active than the starting material that we came from. And it again has hydroxylation of the 5-membered ring. Starting from drostanolone enanthate, we again transformed that using Gibberellum physeroides and obtained a number of products, one of which I've highlighted on the left bottom of the slide, has 2 new hydroxyl groups introduced, and this is more active than the compound that we started from. So a series of these very potent bioactive substances, very simply formed by fungal transformation of potent starting materials to start with, and we were measuring the aromatase inhibition as a result of these experiments. Similarly, XM stain, which is now shown on the slide, was also generated, and then it was transformed into the various products that you have shown on the slide, some of which were very, very active. A number of US patents have been filed on this basis, and we hope that some of these compounds will lead on to clinical trials and something really exciting will emerge. Let me now move to another area in the next part of my lecture. To conclude this part, starting with folklore practices, we have tried to, first of all, look at neurodegenerative diseases and discovered some very exciting molecules against epilepsy, which has led us into dabbling into understanding the mechanisms of thought processes and understanding neurodegenerative diseases. And then again, using biotransformation studies, we have prepared some very potent molecules which are exciting drug leads. In the last part of my talk, what I'd like to spend a few minutes on is just how innovation is transforming economies. I was the Minister for Education, the Minister for Science and Technology, and I also headed the Higher Education Commission for a number of years. And the measures that we introduced in Pakistan to uplift education and science and innovation have resulted in a significant transformation of the landscape of higher education and science in the country. Just to illustrate what has been going on in the world, if you take countries, small countries like Singapore or large countries like China, the story is the same. It's innovation transforming economies. And this slide that you see here is a report that came out from McKinsey Global, which showed that by 2025, it predicted that there'll be $100 trillion impact of a number of new technologies, some disruptive innovations that are transforming economies. And these include artificial intelligence, the Internet of Things, autonomous vehicles, energy storage systems, material sciences, etc. And I gave you the example of Singapore, a small country, population only 5.5 million, and yet the exports were $330 billion last year. And this slide shows how Singapore overtook the United Kingdom in the 1990s in the GDP per capita. Blue line is Singapore, red is UK, and the green is the world average. So in the mid-1990s, Singapore overtook the UK and now has a higher per capita income with almost no natural resources than countries such as Norway, Sweden, Denmark, Germany, or the United Kingdom. And China has also done the same. This slide, in one slide, shows the secret of the development of China, where China started sending a large number of students to top universities across the world. Extreme left is 1970, starting from 1978, the years. The next column is the number of students being sent abroad. So starting from only about 860 students in 1978, China sent about 600,000 students last year to top universities in Europe and USA and other advanced countries. And about 500,000 students returned, were clustered in centers of excellence, and they're transforming China in an amazing manner. So much so that the number of international patents from China today are greater than the number of patents from USA. And China has a higher R&D expenditure also today than the United States. And it's medium and high technologies which is the challenge, the ability to manufacture and export medium and high technology items. And this is what has been happening in those countries which have invested massively in science and innovation. This is the share of, this graph shows the share of manufactured products in world exports by technology. And it is the medium and high technology which dominate. So when I was appointed as the Minister for Science and the Minister for Education and later the Chairman of the Higher Education Commission, we invested major funding in Pakistan, 6,000% increase in development budget of science, and also a 3,500% increase in the development budget of higher education. And we sent about 11,000 students abroad to top universities, started the world's largest Fulbright program. But sending them abroad is just one part. We must attract them back and create the enabling environment for them so that they're able to contribute. So there were several things that we did. Firstly, we brought in a completely new kind of salary structure, where the salary of a professor was made 4 to 5 times the salary of a federal minister in the government. For instance, under the tenure track system, which was a contractual system with appointment for 3 years, international evaluation, then for another 3 years, a second international evaluation before permanency of job was granted. $100,000 research grant being made available to every returning student, for which he or she could apply 1 year before their PhD was completed abroad. Creation of a digital library which provided free access to 65,000 textbooks and 25,000 international journals. A very interesting free access to sophisticated instrumentation. You can't have NMR and mass and X-ray in every lab in the country, but we can share this. And so we brought in a system under which any student, any researcher could get his samples analyzed in any lab in the country free of charge, and the bill for that would be paid by the Higher Education Commission. So the excuse that we can't do this because we don't have that evaporated. and and jobs on arrival. So every student who arrived did not have to waste time looking for jobs. This was done well before his or her return. Quality assurance cells were set up in every university, and a host of other measures were taken, which led to a remarkable transformation of the landscape of higher education. And just to show what happened, this is one example. If you look at the extreme left, these are research publications in impact factor journals per 10 million population, Pakistan versus India. So green is Pakistan, blue is India. So Pakistan had 44 international publications as compared to 172 of India in the year 2000 per 10 million population. But due to the measures that we took to promote research in our universities, this started changing. And as a result of this, the whole, in 2017, this whole picture changed. And by 2017, we had overtaken India, and now we are about 30% ahead of India in terms of research publications in impact factor journals per million population. This is not a small achievement, but it shows that with determination and with suitable policy measures, something really exciting can be achieved. Nature came out with this article in December 2018. We showed that Pakistan had the highest rise in research publications year to year as in the world as compared to other countries. And the chairman of the United Nations Commission on Science, Technology and Development wrote after I had completed my tenure as chairman of the HEC that no other country had shown such spectacular positive developments in the higher education sector than Pakistan in the last 6 years. More recently, in 2016, Thomson Reuters came out with an article entitled Another Brick in the Wall, BRIC, and compared Pakistan to the BRIC countries, Brazil, Russia, India, and China, and concluded that Pakistan has emerged as the country with the highest percentage of highly cited papers compared with the BRIC countries. So something very exciting happened in Pakistan, and as a result, the landscape of our universities has changed very significantly. Interestingly, India became concerned at what was going on at the time, and a presentation was made to the Indian Prime Minister about what I was doing in Pakistan at the Higher Education Commission. And this was reported in the leading Indian daily newspaper, Hindustan Times, in an article entitled Pak Threat to Indian Science. And it started with the words, Pakistan may soon join China in giving India serious competition in science. Well, I think India and Pakistan have to forget their differences and solve them and work together as brothers in arms, because we have to fight together against hunger and poverty. And I hope that we will soon be able to embark on that road of fraternity and brotherhood rather than antagonism. India decided to close down its UGC subsequently. Its University Grants Commission has now established an organization very similar to the Higher Education Commission in Pakistan. It is called the HECI, the Higher Education Commission of India. Before I end, I would like to show a few slides. This is the HEJ Research Institute of Chemistry at the University of Karachi. This is now one of 17 different buildings on 100 acres of land. We have 13 superconducting NMR spectrometers, including this 800 megahertz NMR spectrometer. Many of them are fitted with cryoprobes, which give us increased sensitivity. And these work round the clock of 24 hours, all these instruments, because this is a national center, instrumentation center for the whole country. So we get samples from different institutions across the country, which are analyzed here in our institute. We have about 15 different types of mass spectrometers, which are used for fitted with various bombardment sources. a very interesting pharmacological setup with animal houses. We also have now a nanotechnology laboratory. We have recently set up a virology institute. This is the first National Center for Virology, where we are doing work on COVID virus, and we are culturing virus and looking at drugs which can be effective against treatment of COVID and its variants. I've set up a genomics center in my father's name through a personal donation, which is the state-of-the-art gene sequencing facilities, and we are monitoring the changes in the structure of the virus on a daily basis through identifying the areas where these changes are taking place through sequencing. We have, as I said, many different types of mass spectrometers, as well as pilot plant facilities. And we have a large number of students from abroad because we are the UNESCO Center of Excellence. We are also the WHO Center of Excellence. We have had about 100 German students come to us over the last few years to study and do research here. We also have many, and these are some of the students from Germany standing with me here. We have also many students coming from Africa and from other Asian countries, and we have many joint collaborations going on. We have also set up a large center for nanotechnology, where there's work going on nanopharmaceuticals, among other areas. And the Panjwani Center for Molecular Medicine is focused on some of the new drug research areas, and we have work on stem cells and neuroscience being conducted there. I would like to conclude now by thanking my collaborators, those Professor Iqbal Chaudhry, of course, and I have worked together in these fields, and in the area of discovery of anti-epileptic molecules, Dr. Farzana Shaheen and Dr. Shabana Simji have been involved. In the area of ethnobotanical surveys, biotransformation, and aromatase inhibition, we have Dr. Atiyatul Wahab, Ms. Mavish Siddiqui, and Nimrod Naveed has been involved. Dr. Sama Yusuf has done the X-ray crystallographic studies within our institute. We would like to thank the financial support that we have received from the Searle Company for the Genomics Institute, from the Higher Education Commission, and from the Pakistan Academy of Sciences. It has been a real pleasure for me to be able to deliver this talk. I have been involved in many ASOMPS in the past, and I hope that situation of COVID will improve in the future, and the next ASOMPS will be where we can physically participate, in fact, instead of giving a virtual lecture. So I would like to thank you for your patience, and I hope that this talk that I've given will excite new researchers, both in the area of neurodegenerative diseases, as well as in various more potent aromatase inhibitors, which will lead to cures towards breast cancer. Thank you very much.
Sharing, and thank you Datin Dr. Rohaya Ahmad for moderating the session. Ladies and gentlemen, fellow participants, the time has finally come for our invited lecture sessions A and B. Before that, I would like to invite all participants to watch flash presentation for 30 minutes before entering the respective invited lecture sessions A and B. Please join us again at 3:15 p.m. for the invited lecture sessions A and B. Requests to join the breakout session can be found at the bottom of your screen as well as at the agenda on the website. Hope you enjoy in gaining knowledge and insights throughout this virtual conference. Thank you.
Hello everyone, my name is Norain Fatiha Aldi. I'm a student from Faculty of Applied Science UiTM Shah Alam, majoring in chemistry and minoring in forensic analysis. The title of my presentation is Purinium alkaloid from the Heterostema pipirifolium, a limestone plant species. Heterostema pipirifolium is a climber plant species collected at Pulau Tuba, Langkawi, Malaysia. It comes from Apocynaceae family. This species has lack of information about its traditional uses. However, the aerial parts of genus Heterostema was believed to treat cancer, also act as antiviral and antibacterial. Thus, the objective of this project is to isolate and purify alkaloids from the leaves of H. pipirifolium. For the methodology, the leaves was undergo maceration extraction using hexane and dichloromethane solvents. Then, the dried DCM extract undergo phytochemical screening by Meyer's reagent and showed the presence of cream color precipitate indicated the presence of alkaloid. Acid-base extraction was the best method to extract the alkaloid since alkaloid was the basic compound. After the acidification and basification of DCM fraction, the lower layer containing alkaloid was evaporated to dryness and purified using trituration technique which produced two fractions A and B. Fraction A was subjected to spectroscopic analysis for structural elucidation and characterization. The result of 1D NMR were compared with the data of Heterostema H from the literature review. The difference is only at NH position in H NMR spectrum, where Heterostema H had two broad peaks while alkaloid A only had one broad peak. The peaks of NH in alkaloid A were overlapping to each other. From the mass data, the positive ion mode HRESI mass of alkaloid A exhibited molecular ion at 212.114 and molecular weight C8, H14 and 5O2. This is the structure of alkaloid A with the correlation of COSY and HMBC. Next, the COSY NMR showed the correlation of proton in NH and 9CH3 represents the connection of secondary amine with methyl group. Moving forward to the HMBC NMR that showed the correlation between carbon and hydrogen that was separated by 2 or 3 bonds. This figure showed the correlation between proton in 9CH3 with C4, proton in 7CH3 with C5 and C8, and proton in methoxy group with C6. The correlation revealed the connection of N-methylamino with C4 of the ring, N-methylformamido with C5 of the ring, and methoxy group with C6 of the ring. From all results, it was confirmed that alkaloid A was Heterostema H. As a conclusion, Purinium alkaloid named Heterostema H was successfully isolated from the leaves of H. pipirifolium. For this project, I would like to acknowledge Aurins and Faculty of Applied Sciences UiTM for the facility and opportunity of learning natural product. That's all from me. Thank you.
Assalamualaikum and good morning to the judges and also all the committee members. My name is Nur Haida Shadadamiti from University Putra Malaysia. Today I will present my poster presentation entitled In Vitro Cured Induction of Curcuma cassia as influenced by different formulation of the medium. As introduction, Curcuma cassia belongs to family Zygomeraceae. Curcuma cassia is a rare and underutilized medicinal plant origin from India. This species has been used since an ancient time to treat various ailments and diseases. The rhizomes of Curcuma cassia possess numerous bioactive compounds which traditionally used for the treatment of tonsils, sprains, asthma, diarrhea, cough, jaundice, and epilepsy. In the scientific pharmacological studies conducted, Curcuma cassia extract have a potential as anti-asthma, antimicrobe, anti-cancer, anti-inflammatory, and contain antioxidant properties. In Malaysia, Curcuma cassia is not popular compared to other turmeric species such as Curcuma longa. Hence, Curcuma cassia is hardly available in Malaysia and the price of the fresh rhizome is expensive. As an alternative for conservation of this species, tissue culture technique was introduced in this study for rapid propagation. Methodology: in this study, 3 types of vessel medium were used. The vessel medium used was Murashige and Scope, Udipla media, and also modified MSP5 medium. The vessel medium was supplemented with 5 micromolar BAP, 30 gram per liter of sucrose, 3 gram per liter of gera, and pH adjusted to 5.75. In the base of the results obtained, MSB5 vessel medium was significantly produced the highest percentage of shoot induction, number of shoot, length of shoot, number of leaf, and leaf fresh weight. Meanwhile, WPM medium was significantly produced the lowest values in all parameters recorded. As a conclusion, modified MSP5 vessel medium composition was found to be the most suitable vessel medium for formulation for shoot induction of Curcuma cassia. That's all from me. Thank you.
Chemical profiling of Nittum leptostasiums of fruit extracts using high-resolution LCMS. Nittum leptostasiums is from Nitaceae family, an indigenous plant from Borneo. The plant was collected from Tambunan, Sabah. The fruits are eaten by native people for years. However, there has not been any scientific data on the fruit nor its nutritional value. So we grabbed this gap to study detail on the chemistry of plants otherwise taken for granted. The methanol extract was pre-treated by using SPEC18 to remove the interference. The eluate was dry and prepared at 1 mg per ml concentrations for chemicals and mass profiling using HPLC and LCMS. For the LCMS analysis, the sample was analyzed in optimum condition of positive ionization mode. Dereplication plays a crucial role in natural products, as it can provide fast identifications of non-metabolites in complex mixture. So for these dereplications, EnzyMine software was used as interface for data processing. It was carried out by comparing the full scans mass data obtained from LCMS to costume database created from Dictionary of Natural Products. Out of 186 listed compounds from Dictionary of Natural Products, 47 compounds were identified with mass error less than 5 ppm. Most of the identified compounds come from the class compounds of stilbenes, which consists of dimer and oligostilbene. However, this approach will have tendency to identify more than one compound in one retention time. For example, at the retention time 5.57 minutes, there are about 3 compounds were identified as isomeric compounds because it shares some molecular formula but difference in structure. In the nutshell, stilbene is a major group of compounds in the fruit of Nittum leptostasiums, but the isomeric compound with similar mass requiring further tandem mass analysis. We would like to thank University Technology Mara for GIP grant and Sabah Biodiversity Center for collaborating in this project. Thank you.
Assalamualaikum and a very good day to everyone. I am Nur Hasifi bin Shuib from Malaysia Rubber Board will present the poster with the title Chemical Composition and Phytochemical Screening of Hevea brasiliensis seed. As introduction, oil crop production such as oil palm, soybean and sunflower must increase by 133 million tons in order to fulfill the needs for food, feed and fuel demand. From this, it is forced to look beyond the conventional major oil crop into alternative sources to meet the world growing needs. And one of the potential sources would be the rubber seed. The objective of this study was to determine the chemical composition, mineral contents, oil characteristic, and phytochemical of Hevea rubber seed from one of the elite clone, which is RIM 2001. There are 5 parameters that we account into this study. For proximate analysis, the results show that the rubber seed have high content of moisture, oil, protein, and carbohydrate. Based on the relatively high oil yield, the rubber seed have a big potential for biodiesel production and also can be a good source of protein in human and livestock. There were 5 microminerals and 4 microminerals were detected in the rubber seeds. These minerals are commonly essential nutrients for human body. Besides that, rubber seed contains low acid value and free fatty acid and is desirable for nutritional application. For fatty acid composition, rubber seed contains high level of arachidic and oleic acid, so the rubber seed oil could be expanded to cosmetic industry where oleic acid has been shown beneficial for skin. Phytochemical group of tannin, flavonoid, and terpenoid were detected in the rubber seeds. This secondary metabolite group can be contributed for pharmacological activities such as antioxidant, anti-cancer, and anti-inflammatory properties. Finding for these studies, we can conclude that heavy acid could be considered as an alternative source of oil, protein, and mineral. Heavy acid is able to be used in pharmaceutical industry due to the positive result in phytochemical content.
Assalamualaikum and hi to all. My name is Siti Zafira Zulkifli, a postgraduate student of University Technology Wara. Today I'm going to present my poster with the title of Maize-based identification of Lepisanthes rubiginosa extracts. So basically Lepisanthes rubiginosa, or called as pokok mertajam among Malaysian, is a plant that belongs to Sapidaceae family. This plant is widely used by the peoples as it is deemed capable to treat a variety of ailments such as fever, headache, diarrhea, dysentery, jaundice, and insomnia. It also has a broad range of biological activities, particularly in antimicrobial activity. This plant was primarily researched in 1999, where 12 compounds were successfully isolated. After so long, this species was further studied in the year of 2020, which provide another 4 compounds of saponins, making a total of 16 compounds. So it shows us there is a limited study on phytoconstituents of Lepisanthes rubiginosa, despite the fact that many pharmacological effects have been reported. Thus, the replication technique was implemented to discover more chemical constituents in a short time within the extracts. Replication is a tool to speed up the identification process within the extracts. This work, we use stem bark parts of the plants and extracted them using dichloromethane and methanol solvent. The sample was optimized using UHPLC before subjected to the LCMS with a specific condition. The mass data that we gathered from LCMS were processed and comparing them with previous literature and dictionary of natural products. So here is the total ion chromatogram. As an outcome, a total of 100 chromatogram peaks were successfully detected, of which 32 were tentatively annotated. A variety of chemical class compounds were detected, where 16 were terpenoids, 7 were saponins, and followed by flavonoids, phenolics, amino acid, and butanolide. Terpenoids are most abundant of chemical in dichloromethane compared to the methanol, where saponins are dominant. From the data, we also found that 5 compounds were identical on both extracts, which were highlighted in this chromatogram together with the structure at the right side. In summary, 27 compounds were identified by comparison with database, and the chromatogram revealed more than half of the peaks were not detected, including some major components, highlights more chemical potential can be discovered as a source of new metabolites. Thank you.
Assalamualaikum warahmatullahi wabarakatuh and greeting to our participants. Thank you for being here today. I'm Nur Azul binti Sulaiman from University Sultan Zainal Abidin. Today, I will present my preliminary study on the anti-addiction properties of erythroxylum cornutum against chronic morphine. Erythroxylum cornutum, or known as belungkas bukit, is a big tree natively distributed around Southeast Asia, especially Thailand and Philippines. As for ethnobotanical use, erythroxylum cunetum is used as fish poison in Philippines. There are many fundamental studies were run to observe the effect of erythroxylum cunetum in various aspects, especially in toxicology and ethnopharmacological aspects. This present study was conducted to observe the anti-addiction properties of erythroxylum cunetum on chronic morphine using human neuroblastoma cell line. Cytotoxicity study was run to determine the safe dose of the extract. For addiction study, cells were induced with morphine for 24 hours. After that, the cells were washed with PBS and treated with culture media, which is as control, 5 doses of erythroxylum cunetum and 1 dose of methadone, which is for positive control. The cells were incubated for another 24 hours. The cell lysate were extracted and proceed to ELISA assay to detect the level of the CAMP. As you can see from the graph, the addicted cell treated with culture media show a highest level of CAMP. As per scientifically proven, high level of CAMP indicate addiction. Meanwhile, the addicted cell treated with all doses of erythroxylum cunetum show a normal level of CAMP, which is comparable to methadone. As a conclusion, a colloid extract of erythroxylum cunetum has anti-addiction properties. Again, chronic morphine observed in human neuroblastoma cell line. The effect is comparable or mimic the methadone, which is commonly used in treating addiction. However, more study must be conducted to conclude the effectiveness of this plant. I hope by doing this preliminary study, it will open a new opportunity for another alternative medicine in treating addiction. Thank you.
Hi everyone, welcome to 17th Asian Symposium on Medicinal Plants and Spices. Ladies and gentlemen, for the past 30 years, plant cell and tissue culture technology has been used to produce several valuable phytochemicals for the pharmaceutical and nutraceutical applications. So we have conducted a study to evaluate the total phytochemical contents between in vivo and also in vitro planters in this important medicinal plant called Thalinum fruticosum. This underutilized herb, also known as Ceylon spinach or Javanese ginseng, is traditionally used as an elementary tonic and consumed as vegetable leaf. It also possesses various pharmaceutical properties due to high flavonoid and saponin content. However, the susceptibility to various diseases has limited the mass cultivation of this species in the field for the industrial application. Therefore, we believe tissue culture technology could provide a sustainable alternative sources of the plant products for this species. Basically, the idea of this study is to evaluate is there any significant difference of the total phytochemical contents between the field-grown plants and in vitro plantlets. In this study, the 4 months old of field-grown plants and in vitro plantlets were used and was extracted through methanolic extraction. To quantify the total phytochemical contents consists of phenolic, flavonoid, and saponin through photometric quantification. Our result shows the higher total phytochemical was recorded in the in vivo leaf consists of the total phenolic, flavonoid, and saponin content. However, there's no significant difference of total flavonoid content between in vivo stem and also in vitro plantlets. Furthermore, the total saponin content was found higher in the in vitro plantlets compared to in vivo stem. Based on this preliminary study, we suggest that the in vitro plantlets could provide a sustainable alternative sources of flavonoid and saponin for the industrial application. We could use biotechnological approach, for example, secondary metabolite engineering to enhance the production of secondary metabolites in this species. With that, I would like to thank to my institution, International University of Malaya Wales, for providing the grant to conduct this research. Thank you so much.